and today we are going to start with plant tissue culture applications. So if you are doing your first class, even then you are able to uh, understand what I am going to explain. So first thing, what is plant tissue culture? Plant tissue culture is a technique in which we can grow a plant in lab conditions. When we are doing any technique in lab condition, that is known as in vitro technique or in artificial techniques. So here, as you can see, these plants are growing in a flask, or you can grow them in a test tube also, or you can grow them on a petri plate also. And here you can see these different developmental stages of a plant. You took this explant, it is very small here. Slowly and gradually it will start growing, and after that it will grow in the size. And after that it has well developed shoots and roots, then we can transfer this to the field condition. That is what all plant tissue culture is about. These are the some applications of plant tissue culture. Here we can do the development of superior cultivars. What do you understand by superior cultivars? Cultivars are the varieties which farmers are growing in their field, and we want these cultivars to be superior in every aspect, superior in yield, superior in quality, without any disease, without any insect attack. So they should have disease resistance and insect resistance also. With the help of this technique, we can do germplasm storage. We have a separate chapter on germplasm storage that we will be covering in detail in our syllabus. Germplasm is sum total of genes available of any plant species. For example, if you take a plant, for example, bamboo is there, and you just travel all over the world and you collect all the species of bamboo which are present in entire world today, and you make a collection of that collection will be known as germplasm. So in vegetatively propagated crops, it is difficult to store these cuttings in field conditions. That will require a lot of things: land, labor, there will be climatic conditions. So how can you store that germplasm, those bamboo plants? You can store them in artificial conditions. You can grow them in flasks and then store them on a shelf. That's how plant tissue culture helps you for germplasm storage. Then second application here is soma-clonal variations. Soma-clonal variations are those variations which arise in plant tissue culture raised plants. When you are doing tissue culture, you are taking an explant and you are making a number of plants from that explant. So what do you expect genetically? Genetically, you expect that all plants will be similar. That's how they are known as clones. But in tissue culture, when you are doing it, due to differences in medium composition, there may be slight differences in temperature conditions. So due to these reasons, sometimes some variations arises in tissue culture raised plants. And these variations may be different. Obviously, that's why they are known as variations, and they may be beneficial too. So if such thing happens, such they are known as soma-clonal variations. Next application is embryo rescue. In some plants, it happens that embryo is formed after pollination, fertilization. Embryo is formed, but that embryo does not develop a fully ripened seed due to abortion of embryo, because there are sometimes post-fertilization barriers in some plant species. So in such species, you can help that embryo in its growth by transferring it onto a nutrition medium where it can grow into a plant. Ovule and ovary culture, anther and pollen culture, these both type of cultures are conducted to get what you want to get from here, haploid plants. These both, these will give you haploid plants. Just one condition is that this ovary should be unfertilized, because these are gametic tissue of the plant. So when you are culturing these gametic tissues which are haploid in nature, so you will get haploid plants. So if you want to raise haploid plants, it's simple. What you can do, you can culture anther pollen or ovule or ovary that is in non-fertilized condition. Next is callus and protoplast culture. So these are two things. Callus is a actively dividing undifferentiated cell mass, where you have put your explant on oxygen rich medium, mostly 2,4-D, 2,4-dichlorophenoxyacetic acid, that is an oxygen. It is added into the nutrition medium. Explant is put on 2,4-D medium, and then what happens? There is very rapid cell division that is initiated in the cells of explant, and those cells are just dividing. That's why I am saying actively dividing cells, and they are forming a non-differentiated cell mass that is known as callus. And from the cells of the callus, you can isolate protoplast, which can be cultured also. So what is protoplast? Protoplast is a cell without protoplast. And these protoplasts are fused to make somatic hybrids, where normal hybridization is not possible due to various reasons. You can do protoplast fusion. You will take protoplast of two species, species A and species B, and you can fuse them in lab. That's why you can generate a hybrid by the protoplasmic fusion. And such hybrid which has been obtained by the fusion of protoplast of two different species is known as a somatic hybrid. Then with the help of tissue culture, you can screen a plant in vitro. Screening means selection, selection for different traits. Whether your plant is having insect resistance, your plant is having disease resistance, it is having certain quality values. So for that, selection can be done in vitro, that means in lab condition, if your plant is growing in lab. And last but not the least, you can do multiplication with the help of tissue culture. With the help of tissue culture, we are making a lot of planting material that is known as high quality planting material with the help of tissue culture. Why it is high quality? Because it is disease free, pathogen free and virus free. So these are the techniques which you can perform in plants with the help of tissue culture. One is micropropagation, one is germplasm preservation or germplasm conservation, another is somaclonal variation, one is haploid and dihaploid production, and last is protoplast fusion, that is somatic hybridization. On each of these applications, we have a separate chapter and we will be discussing them in detail one by one in our coming lecture. So you have to study about micropropagation, germplant conservation, somaclonal variations, haploid productions, and protoplast fusion. So this is micropropagation. Micropropagation, as the name says, it is of two terms. One is micro and second is propagation. So what do you understand by propagation? Propagation means multiplication of plants, how you cultivate the plant. So there are basically two methods of plant cultivation. Your plants may be cultivated either by seeds or by vegetative means. So when you are producing plants by seeds, that is known as seed cultivation or sexual reproduction of the plants. If your plant is not producing viable seeds or seeds are having other problems, then like dormancy is there, some inhibitors are present in the seed, then what can you do? You can produce some plants by their vegetative means. There are different vegetative means like there is grafting, cutting, air layering. So these are different methods. So when you are growing a vegetatively propagated plant in tissue culture methods, that is known as micropropagation. Very short definition of micropropagation is in vitro clonal propagation. Clonal means when the offspring plants are genetically similar to the mother plants. So when you are doing vegetative propagation, it always happens that your offspring plants, multiplied plants, are always similar to the mother plant. So that's why they are known as clones, and this method is known as clonal propagation. And then the same technique is being done in lab. It is known as in vitro clonal propagation or micropropagation. So this is one method of micropropagation which is known as meristem tip culture. So what is meristem here? Meristem is the most uppermost tip of the plant, the growing tip of the plant. Why it is taken basically in case of micropropagation? Because in vegetatively propagated plants, why it is need to grow them in lab when you just can grow them in field by inserting a cutting in the soil? You just make a cutting of the plant, insert it into the soil, it will root and shoot, and you will have a new plant. Then what is the need of in vitro methods? What is the most posing challenge in vegetatively propagated plants is the infection or attack of pathogens, attack of viruses and disease incidents. Why so? For example, your vegetatively crops like potato is there, bamboo is there, and sugar cane is there. So how farmers multiply it? For example, sugar cane. So its nodal segments are taken as planting material. When these nodal segments are being cut from the mother plant, so plant for cutting, the farmers are using the same instrument, the same sickle. So if a plant is having any infection, any pathogen inoculum is present on a plant, when all the plant material is being cut by the same sickle, what will happen? The pathogen inoculum will spread to all the planting material. And if pathogens are present in soil at a place when you are doing soil operations, what will happen? With the help of your machinery, that pathogen will spread to whole of the field. So such thing happens. So to avoid these things to spread, what you are doing now, you are taking this uppermost part of the plant which is known as meristem, and this part is cut from here. You make a cut here and this part is excised. Then you will remove these older leaves and you have this meristem. This is uppermost growing tip. Since it is very new, so it is free from pathogens, free from infection. And what will you do now? This apical meristem is excised. After that, this is sterilized. What is sterilization? That means it is treated with some chemicals to make it free from the microbes. So it is inoculation onto nutrition medium. After inoculation onto nutrition medium, it will grow into shoots and roots, and that's how you have a bigger plant, growing plant, which is transferred to the pot. So that's how we do the micro propagation by meristem tip culture. Why we do micro propagation? Because here, what is the advantage? Here you can make very easily one to many. You are taking a small tiny explant, and that small tiny explant can make here many plants. Whereas in field conditions, when you are multiplying plants from cuttings, you will get only one plant from one cutting. But in lab conditions, you can raise a number of plants from one explant. Then second advantage, multiplication in controlled laboratory conditions. So when there is controlled conditions, that means here you can control temperature, you can control photo period, you can control humidity also. So everything is in your control. There won't be any effect on the plant growth due to climatic condition. Whereas in case of field condition, weather conditions may be extreme. Sometimes the temperature goes very high, as in summers, and where in winter crops the temperature goes very low. There may be flooding. So such calamities, environmental calamities can be there. These are unavoidable, whereas in lab conditions, when we are doing micro propagation, such things can be avoided easily. Then continuous propagation year around. That is again one more advantage of micro propagation, that you have your plant material, ready plant material the whole year because there is no environmental constraint. Then you have all plant material which is disease free. That is one requisite in plant tissue culture. If disease pathogen is present there, it will not let your plant grow. So if your plant is growing, so it is disease free. Inexpensive per plant once established. So all the efforts goes into the establishment. Once your cultures are established and your growth cycle has started of that plant, then you have planting material in enough quantity and the cost will be highly effective. Precise crop production scheduling can be done when you are doing micro propagation. And you can reduce stock plant space because your plants are in very small containers. So you can easily stock them up wherever you want to. So since this is a very good technique, still it has some limitations. So what limitations are present in micropropagation? You require everything here to be specialized. Your equipment should be specialized and your facilities which you require for doing tissue culture, they also should be specialized. And you require here to perform micropropagation, that is technical expertise. Technical expertise will be required. No layman just come and perform micropropagation. It is not possible. That's why technical expertise will be required. A skilled person rather, who has earlier done tissue culture will be preferred. Protocols not optimized for all species, since all the species have not undergone the tissue culture process. So sometimes it may happen that the species you are working with, you have to optimize the protocol for that species also. Protocol optimization means what medium you are taking, what should be the concentration of growth regulators, what should be the sterilizing agent, what should be the duration of sterilization. So each and everything you need to optimize according to your species on which you are working. Okay. Plant products may not fit industry standards. Sometimes it may happen, whatever you are producing, the time of production and the quality of production may not fit according to industry standards. Relatively expensive to set up. When you are setting up tissue culture laboratory, it will be relatively expensive. Relatively to what? Where you grow your plants? To the fields. If you have a field, your own field, and you are growing these plants in lab conditions, then it will be comparatively expensive. So what are the applications of micropropagation? By the help of micropropagation, you can do rapid increase of stock of new varieties. There is elimination of diseases, cloning of plant types not easily propagated by conventional methods like some offshoots, sprout, seed, date palm, phones, etc. Then propagules have enhanced growth features. These are the methods of micropropagation, how micropropagation is done in lab. When you are doing micropropagation in lab for a plant species, you can go for axillary branching or adventitious shoot formation or somatic embryogenesis. So axillary branching is also known as axillary bud proliferation. Here the explant used is axillary bud, which is present in the axil of leaf. And where is the axil of leaf? Axil of leaf is that part of plant where the leaf is attached to the stem. If you see very closely, then in the group which is made in the leaf, there is a axillary bud that is in dormant conditions. But if you grow them on nutrition medium, their dormancy will go away and they will start to proliferate into a plant. So you can do axillary branching. Almost more than 95% of all micropropagation is done by this method. Then adventitious shoot formation, that is organogenesis. You are growing a shoot from such plant part which is not made for making shoots. So that's why it is known as adventitious. And last one is the somatic embryogenesis, in which you make somatic embryos which are mostly likely to zygotic embryos. So this is axillary shoot proliferation. Here you can see the axil of the leaf. This is the axil. These are known as axillary bud. So growth of axillary buds stimulated by cytokinin treatment, because you are raising the shoots. So these shoots arise from this meristem, and these are known as axillary buds. Clonal in vitro propagation by repeated enhanced formation of axillary shoots from shoot tips or lateral meristem Cultured on media supplemented with plant growth regulators, usually cytokinin. Shoots produced are either rooted first in vitro or they are acclimatized ex vitro. One more important slide. These are the steps of micropropagation. So first is stage 0, that is selection and preparation of the mother plant. So selection criteria of the mother plant, we have already discussed that mother plant should be the elite class. Elite class means it should be the best of the lot. So how will you select it? It is selected based on the visual characteristic or morphology. So that is the criteria of selection. It should be an ideal plant. So it should be high in height. It should contain higher biomass. Yield should be high, and it should have insect and disease resistance. Disease free should be preferred. And after that, this plant should not be sprayed with any chemicals 14 days prior to explant collection. That's what you have to keep in mind whenever you are going for selection and preparation of the mother plant. Then from this plant, you will take the explant. So this is the last slide we are going to discuss. After that, we will do the quiz part. Then comes the stage 1. After you have done the surface sterilization of the explant to make it sure that your explants are free from any pathogens, you will do that. So stage 1 will be initiation of culture. In that initiation, that washed and sterilized explant is capped onto nutrition medium, and that is known as initiation of culture. Then stage 2 is known as multiplication. Explants transfer to shoot media. Shoots can be constantly divided. So what is shoot media? Shoot media is the basic nutrition medium which is rich in MS medium plus cytokinins. So with cytokinins, it may have BAP, benzyl aminopurine or benzyl adenine, kinetin or zeatin. Then shoots are constantly dividing. And we can get a number of plants from the single explant. After that, stage 3 would be rooting stage. In the rooting stage, the medium which is used is known as RIM, that is root induction medium. And at this stage, oxygen concentration should be higher. In shoot induction medium, the oxygen cytokinin ratio is 1:10. That means if you are taking 10 parts of the cytokinin, oxygen should not be more than 1 part. You can use either cytokinin alone or if you are using in combination. So always remember the ratio should not go higher than 1:10, oxygen to cytokinin ratio. So same here in case of root induction medium, this ratio would be reversed. That means it would be 10:1. Oxygen would be 10 part and cytokinin should not be more than 1 part. So that is the main manipulation of tissue culture, that formation of shoot and root. It happens with the change in the medium concentration. Basic concentrations would be same, mainly change would be in the hormonal concentrations. When you have done this transfer on root media, rooting will be there. Then last stage is transfer to soil, where you are doing hardening and acclimatization. So for hardening and acclimatization, what will happen? The plants which you have raised here in tissue culture, you will take them off the flask. you will wash the roots what you have developed. There should not be agar media left, and then this plant is sterilized into a first in a disposable pot. You keep it in your culture room, then transfer to the greenhouse, and once it is growing well in greenhouse, then you can transfer it into the field. That's how you do the acclimatization or hardening of a plant in tissue culture. So these are the steps of introduction explant to the nutrition medium, that is known as establishment of culture. Then you will do the shoot induction, that is known as multiplication and proliferation. Then you will do rooting, and last stage 4 is known as acclimatization. So these are the 4 stages which we have discussed. So if you have any question, you can ask the questions now. Rest of the lecture we will do tomorrow. Questions? If you have any question, just ask the questions. That was all about the plant tissue culture applications from my side for the day. Rest we will discuss tomorrow. For listening carefully, thank you so much. And now we will start our question answer session. Thank you so much for listening carefully.